My name is Shalanda Julakam, the host today. Today we're going to have a seminar on the topic of game-changing material and semiconductor analysis with ICSPI's AFM on a chip technology. Let me introduce you a little bit about DKSH. Our DKSH Business Unit Technology is a leading provider of scientific solutions across Asia Pacific, and we support the growth of our partners' scientific applications by providing distribution, expertise, service, and training. DKSH bridges global innovation with local expertise, helping researchers and laboratories in the region access the world's most advanced technologies. We're exclusive distributor for ICSPI in 3 countries, Philippines, Thailand, and Vietnam. And our partner today, ICSPI. ICSPI is a Canadian company specializing in accessible desktop atomic force microscopes, or AFM, by using advanced integrated sensor technology. They offer systems like nGauge and Redux that simplify nanoscale measurements, providing scientists and engineers with fast, easy-to-use tools to accelerate discovery. And our value speakers today, Dr. Robert Meagley, he's a vice president of quality from Tandem PV, and Xin Shu, an application engineer from ICSPI. For Xianshu Wang, he's graduated from University of Waterloo in Canada with a background in nanotechnology, engineering, and time spent in semiconductor and display industries. He has excellent understanding of challenges and applications involving material science and the current struggles of researchers. Currently, he works as an application engineer and serves as an expert on ICSPI desktop MEMS AFM technology. In his spare time, he enjoys baking and Muay Thai. And our second speaker, Dr. Robert Meagley, he's a leading expert in the design and synthesis of small molecules and complex molecular systems, recognized for his impactful career at organizations like Intel and the US Department of Energy. A prolific innovator, he holds over 41 patents and has authored more than 38 technical papers in his field. His deep expertise span crucial area for material science and the semiconductor space. He's a respected thought leader, providing consulting and lecturing services on technology, nanotechnology, MEMS, and biotechnology to diverse professional community. So I think you are ready. Let's get started with AFM on a chip technology by Xianshu. Xianshu, the floor is yours.
Amazing. Thank you so much, Chalanda. Thank you so much for your kind introductions, and I'm very excited to present to everyone today some introductions regarding what we do at ICSPI and to give a basic overview and rundown of our technology. So let me take over sharing my screen here. I will share now. Here we go. All right, everyone should be able to see. Please let me know if there's any issues with this. But if there's no issues, I'm happy to proceed ahead. So thank you so much, everyone, for attending this webinar. I'm very excited to present to you our AFM technology, our instruments, and how they can support material and semiconductor analysis. So let's continue. So going forward for the introduction, the wonderful introduction that Chalanda made for us, my name is Chinshu Wang. So Chalanda's already mentioned, my personal background involves studying nanotechnology engineering at the University of Waterloo in Canada. I spent some time working in industry as well, working with flat panel displays and the semiconductor industry as well. Now, this is also a photo of me on the right from my recent trip to Thailand a couple of years ago. And this is from me going to train Muay Thai, actually, so Thai kickboxing. Now, going on to our company, so ICSPI. ICSPI is a team of 10 scientists and engineers coming out of the University of Waterloo. As Chalanda mentioned, we were founded in 2007. We've been selling commercial systems since 2017, and we have instruments in over 40 different countries. We were originally funded by DARPA, and we're on a mission to bring powerful, intuitive nanoscale imaging tools to your benchtop to support researchers. How do we do this? We do this with our AFM on a chip technology. Now, on the left-hand side here, you will see a SEM image actually of our MEMS AFM chip technology. I understand that it looks relatively complicated, but I'm going to go into detail on this later. So please save your questions until the end. And this is going to be a really good breakdown of the technology, and I'm going to explain how it works. We have many systems all across the world and in various types of institutions and organizations. We have systems at various United States national labs, at all levels of universities, different colleges, and at a variety of companies ranging from industry, from like established industry players to startups. So let's continue. Our two instruments that we are selling right now are the Redux AFM as well as the nGauge AFM. I will go into some more detail about this later as well. And this won't be a presentation primarily focused too much on these instruments. I'll be talking a lot about our technology, our on-chip AFM technology, and how it works. But this is just to let people kind of get an idea and see what our instruments look like. And we can answer any questions as needed. This is an example of some of our customers. So we have customers here such as Boeing, 3M, and various universities all across the world as well. Now, moving forward, this is a list of some recent publications that researchers have published. And these are publications that use our AFM technology to gather atomic force microscope images in a variety of different research fields. Some of these include electrochemistry research. Some of this involves like membranes research, polymer research, and there's a whole wide gamut of research that our applications that our instruments can support. Now, on to the next point here. It's first very important to talk about what exactly is atomic force microscopy. I know that not everyone here in this webinar is going to have a solid background in what AFM is. So I'm happy to introduce the technique to people and kind of give a rundown of how it works. So AFM is a surface profiling technique. It captures 3D surface images at the nano and micro scale. Effectively, it uses a sharp probe tip on the end of a cantilever to physically scan across a sample. As you can see here on the left, as that cantilever encounters obstacles, either peaks or valleys, it's going to deflect up and down, and any of those perturbations are going to be recorded as signal, which is then used to reconstruct the image. A joke I like to make with people sometimes is that since ancient times, humans have learned things about other things by poking them with sharp sticks. We still learn things this way, except the sticks we use are much sharper and they are much smaller. AFM is especially known for being able to collect very high quality quantitative topographical data, as well as other related information. So, talking about some background of what AFM is. So AFM as a technique was invented in 1985. Traditionally, AFM relies on a system of lasers and photodiodes coupled with an XYZ piezoelectric scanner. Typically, what needs to happen with AFM is that a laser must be aligned to the cantilever itself. There's going to be a mirror on the end of it, and then that has to be aligned to a photodiode. Any changes in that laser signal are what is used to actually measure any deflections of that tip on the sample. As well, the tip itself is not moving. It is the sample itself on a piezoelectric scanner. I am not a major expert in traditional AFM, so I'm happy to take any comments or questions or any points from others with more experience. And Rob might talk about this a little bit more as well later. What are some of the major advantages of atomic force microscopy? One of the major ones is that AFM can give you sub-nano and sub-angstrom resolution. It is a nanoscale imaging technique. It also gives you very powerful quantitative 3D data. With AFM, you are not limited by the diffraction limit of light, like you would be for optical microscopy. And because AFM is a physical stylus technique, you're able to get very precise information on how tall certain features are. You're able to really get very in-depth information and a lot of statistical information. AFM also supports a variety of imaging modes, which I will talk about later as well. AFM also has a lot of sample and environmental flexibility. So compared to something like electron microscopy, With AFM, you do not need to coat any non-conductive samples to make them imageable. You do not need to do any sort of vacuum pump down as well. On the left-hand side here, you'll see an image of a scan taken with AFM. This is of a copolymer, and you'll notice that this scan is approximately 15 by 15 microns, and that the total range of Z values ranges from -37 to 126 nanometers. Now, going back a little bit to traditional AFM and some of the issues with it and what we've really tried to improve on. Traditional AFM can often be very complicated. They often involve very large systems with a large amount of physical overhead, such as the laser, the piezoelectric scanner, and it can be very difficult for a user to actually get an AFM set up to take good scans. They often are limited in terms of where they can be stored as well. They're often very noise sensitive, and they have to be stored in the metrology bays of basements of buildings. And they often are just very challenging to use, mostly due to the laser alignment issue. This is an article written by a very prominent AFM researcher, Dalia Yablon, talking about how even though she has 30 years of experience working with AFM, it can still be a challenging and frustrating technique to use for a lot of the reasons that I mentioned before. She talks about what needs to be done to make it more user-friendly and accessible on a physical level. Fundamentally, what we are trying to do as an organization is to try and resolve this to make such a powerful technique more accessible to people. Ultimately. So our goal here is to make a powerful technique more accessible. You can see some of our on-chip AFM on the left. And again, I'll explain more of that as we go on to the next slide. So ultimately, the purpose of my talk here is to go over AFM kind of as an introduction to make sure people understand what it is that we do, what AFM can provide for people, and discuss how it can be underused today, even though it's very powerful. Our chips look complicated, but they greatly simplify operation of AFM, and we've ultimately made AFM much easier and faster to use with the ability to scan in only a few minutes. So talking a bit now about our AFM on a chip technology. What we've been able to do is that we have taken the functions of AFM and we have miniaturized it onto a microelectromechanical or MEMS chip. This greatly simplifies usage and operation, and we've automated a lot of the process of AFM with integrated sensors. So for instance, we do not need to do any sort of laser alignment for our AFM. Because we have sensors built into the AFM probe itself, our probe is able to self-sense how far it is away from certain samples. So you don't need to rely on an indirect sensing method like aligning an optical laser with a photodiode. We also do not need a piezoelectric scanner, so this eliminates issues regarding piezoelectric drift. Our AFM units are controlled using XYZ actuators built into the actual AFM chip itself. This has allowed us to cut down a lot of the overhead that traditional AFM typically requires, and it makes our systems much, much easier to use. And the other thing as well is that because our systems are smaller, they're a lot less noise sensitive. Traditionally, AFM is has a lot of issues regarding noise because they are so large. They're heavily influenced by low frequency noise, but our systems reject a lot of them. So kind of talking about it a bit here, this is a close-up. This is an SEM image of one of our probe tips on the end of a cantilever. This is a diamond-like carbon tip with a tip radius of less than 20 nanometers. We sell a variety of different probe tips for different applications. And here, this talks a little bit about how our AFMs are significantly more noise resistant because they are so lightweight. They automatically reject a lot of low-frequency noise. And we also have very flexible systems that work on a wide variety of samples, which I will talk about a little bit, and then Rob will talk about as well. So this is an example here of our AFM MEMS chip loaded onto a carrier PCB. So what we end up having is that we have a chip that is actually able to be picked up and moved by hand and loaded into our systems. Traditionally, AFM probes are much, much smaller than this. They're about the size of a grain of rice, and they'd be carefully loaded in by tweezers. And we've made a system that's much more user accessible. So this is also another really good slide here that talks a little bit about our probe tips for AFM. Traditionally, AFM probe tips will usually last you about 1 to 2 dozen scans, and they typically blunt out. So because of the contact forces involved, they essentially get too blunt to be able to image well. Now, shown here, this is one of our AFM probe lifetime tests. So we're scanning 22 nanometer features on an Intel processor. These scans are taken over 72 hours, and you'll see in here that the number here is incrementing. So this is the total number of scans. You're able to see that we managed to take up to almost 1,500 scans, and we see no dilation of features. Typically what happens is that when you start having issues regarding your probe tip blunting out, you're going to see the actual features you're seeing slowly get wider and wider because the probe tip itself is getting blunter. We see here that we take these scans over 72 hours, and we see none of that dilation. Now, what are some of the major capabilities of AFM? I'm going to go over this in a little bit of detail, and then Rob is going to get into this with regards to specific applications more as well. AFM, fundamentally, our AFMs, AFM can do many things. So when you have AFM, you can do things like apply a current or apply like a temperature change to a tip. Our AFMs primarily focus on 5 different applications. So we have topography, roughness, looking at step height or film thicknesses, doing particle analysis, and phase imaging as well. So moving on to the first application, AFM is just very useful for looking at anything that can be challenging to image, obviously with optical, or even if you're using electron microscopy and you're not able to get very good data or info about what it is you're looking at. So in here, you can see a 3D scan. This is a topographical scan of the pits on a DVD. And we're able to see these pits, which encode the data within within a DVD itself. And we're able to zoom in on these individual pits and clearly measure how tall each of them are. Another great example here is that one of our customers in Denmark, AKSPI, they are a company that produces metal polishing blends. Previously, they were using an optical microscope to try and assess the depth of scratches left behind after creating a blend and using it to polish metals. They want to get a sense of how deep these scratches are, what is the roughness left behind after the process, and they want to be able to use this data to further hone and develop their processes for developing these polishing blends. So using our AFM, they're able to get actual information about how deep these scratches go, and they're able to get numerical data instead of just an idea based on the picture that they were previously getting out of optical microscopy. So talking about this further, this is the same thing. This is an example of a polished titanium alloy. So here you can see a 3D representation of the topography on the left, and on the right hand, you can see various statistical quantities that come out with AFM. You're able to see that the RA roughness is approximately 16.85 nanometers. You're able to get information on the maximum, on the minimum for pit depth and peak height. You also get able to get information such as surface area. AFM allows you to get very powerful, very precise data that then allows you to further hone your processes. And AFM is a really good tool for assessing the effect of processes on different materials and doing before and after comparisons. This is another example right here. This is a microfabricated pattern on silicon. This is done at the University of Waterloo at a microfabrication facility. On the left hand here, you're able to see a top-down image taken using a scanning electron microscope. And we're able to get a very good image. However, we don't really have a good sense of how tall some of these features are. Now, I know that SEM, you're able to take cross-sectional images, but it can still be challenging for a variety of reasons. AFM has much lower sample requirements, and you're able to get that data fast and easily. This is another example right here. AFM can be used to measure specific dimensions and thicknesses for microfabricated structures. This is another one of our customers in Canada, and they are using their nGauge AFM to accurately measure the thickness of these gold ticks as they grow and e-beam resist. They could not do this before, and this allows them to have much greater control over their processes. The other aspect that AFM is really good for is looking at particles. Now, I'm sure many people here are familiar with various particle size analysis techniques and DLS. These are very good for giving you an idea of what is the effective size of particles. AFM can also give this information, but besides that, AFM can also give you a sense of what is the particle size, what is the particle size distribution, as well as how these particles are distributed, as well as the specifics of their particle shape. You can see here on the left side, we're able to see the 60 nanometer gold nanoparticles. And for instance, this can be compared to other techniques like tunneling electron microscopy, which is commonly used for particle analysis, but can often be very expensive and it can involve like a much higher investment. whereas AFM, for much less preparatory requirements, is able to let you see essentially similar detail to that. Now, let's go forward. The other thing I want to show here is a silica polymer composite. This is provided to us by an advanced materials company in the US. On the left-hand side, you'll see a topographical image. On the right hand, you will see a phase image. This silica polymer composite involves a hard silica nanoparticles embedded within a softer polymer matrix. Our AFM produces phase images. What phase images are is, because our AFM operates in tapping mode, there's different modes available for AFM that I can talk about as well, but essentially our AFM is constantly tapping upon the surface of a sample. When it encounters softer areas, there's more hysteresis, there's more sticking. When it encounters harder areas, there is a harsher, stronger bounce back. That phase difference is able to be picked up, and using that, we're able to get a qualitative sense of hardness of materials. On the right hand, you'll see that the hard silica particles, which may be hard to see in the topographical image, show up much more clearly now as the dark red areas compared to the blue polymer matrix area. This can be very useful for people that are building composite materials, that are trying to mix two materials together. They have an idea of what is the proportions of each material relative to each other going in. They want to get an idea of how well these materials are distributed. They want to get an idea of, like, okay, what kind of structures are coming out? Are my final resulting distributions kind of what I'm looking for? Another example here is looking at nano imprint lithography. This is for a company called Smart Material Solutions in the US. They previously were using SEM, but they needed to characterize defects, and SEM wasn't effective for them. So they're essentially working on these nano imprint lithography masters, as well as the imprinted molds that come out of it. Using our AFM, we're able to see very precise information about the heights of various structures and features, as well as get statistical quantities that you can see on the right hand here. And continuing on, same thing. This company wanted to find defects. They were circled in red on the image on the right, on their NIL molds and some of the resultant patterns. The reason SEM wasn't working for them is that they were seeing too much charging. They couldn't coat their sample. They weren't able to get numerical data as well. So here we were able to use AFM, and we're able to pull out these line profiles. So whenever you take a 3D AFM image, you're able to draw lines across that scan and receive 2D line profile information that you can carefully look at yourself to check for specific metrics that you're looking out for. So in this case, we're looking for particle contamination and that the imprinted structure fits the specifications as shown on the left, or let's see particles on the mold as well. Another example here, this is at the University of Waterloo. I actually paid a visit to the lab of Professor Jeff Gostick, and I helped him investigate microporous layer coatings on titanium cloth. Characterizing roughness is important as a function of MPL loading. We just visited in the morning, and previously they were using SEM and optical profiling, but they weren't really revealing the roughnesses that they needed, but AFM did. The roughness can impact catalyst layer and affect performance of the cell. We're able to take these scans using an instrument that we brought over. This was all done in a couple hours in the morning, and I ended up teaching his grad students how to use the tool by the end of that trip as well. A recent visit also was to another local university at Western University in Canada as well. I was visiting Professor Yang Zhao. He was very curious to see lithium samples actually using AFM. He needed to look at these lithium samples within glove box. He wanted to see and compare coated versus bare lithium samples. And he wanted to test and see what the effects of electrical cycling were on these samples and comparing the performance of a coating. So we were able to set up our AFM, our nGauge AFM, inside the glove box and to take these images. So these were done all in an argon atmosphere glove box. And I was even able to teach his research group how to use the AFM as well in a single afternoon. Now, I think that should be the end of my slides, actually. And thank you so much for everyone's patience and for everyone listening to me. And I hope that people learned a little bit about our technology and AFM and the usefulness of this for research. Again, my point here is to just give an overview of the basic technology and to give people some ideas and some examples of how it can be applied to various research applications. Thank you so much. We can see your presentation now.
Yeah.
So I think
Okay.
I'm terribly sorry about that. Don't know what happened, but I'm glad it's all resolved.
Mhm.
Hopefully you can hear me just fine.
Yes, very clear. Please go
Very
ahead.
good. Well, thank you very much. I appreciate the opportunity to present this morning. I'm going to be discussing several aspects of using the ICSPI nGauge AFM in my work in electro-optical materials and semiconductors. I've been a very proud owner of the nGauge instrument since 2017, the year they started commercial release of this. Um, but you can see here in the title slide a little collection of my microscopes. I have here from approximately 1925, actually a little bit earlier, a Leitz bacteriological microscope that's equipped with an oil immersion lens with an ultimate resolution just a little bit more than a half a micron if you really push it. We've got from the mid 20th century a binocular dissecting microscope by Nikon. A couple of digital microscopes. I'm using one of them as an eyepiece for the Leitz. But then down here, small, unassuming, but remarkably powerful, is the ICSPI nGauge. Roughly 100 years separate these two instruments, and it's more than 3 orders of magnitude in resolving power. With the nGauge, it truly will provide nanometer scale resolution as a matter of routine, and I've come to rely on this tool in my work. On the screen over here, you can see some pictures that we'll revisit a little bit later. Let me go forward here. I think one of the most important aspects of the success with this instrument is being able to first pack it up and carry it quickly with you. I've loaded them in its little case and put it in my luggage without further care, gone across the country, unloaded it, and been up and running samples at the work site within an hour. And some of that time is, just basically finding a space to work. One of the things that I've found that's very useful is providing a wind break. A small cover can be had very economically. We've got a couple of different transparent containers you can see that I've used. This one sits in my home lab, and honestly, for convenience of transport, I usually get something on site. They're very inexpensive, but they provide plenty of protection, both from the wind and also from curious observers. You definitely don't want someone touching the instrument while it's running. This instrument here is resting on a granite slab. That granite slab is sitting on a small piece of foam. You can see in the middle picture a very similar circumstance. On the right, though, we've got somewhat more sophisticated vibration control where it's sitting on a specially designed table. This particular table isn't plugged in. It's acting just as a table, and we've got the granite slab on the little foam pad standard, and it does a remarkable job. Every image that you see in my presentation comes from that very simple vibration control strategy that's found very effective for more than a decade. Um, Ultimately, your ability to control the environment will really help your measurement quality. You want to keep air away. You want to, protect it from gross vibration and meddling from bystanders. It's an object of curiosity, so people definitely will want to hear about the tool. But while it's running, you want to make sure that nobody, bumps the table or, upsets the tool. A word or two in the user's manual. We'll talk about proper mounting of the AFM chip. You can see the chip is sitting here mounted to the platform. But they also mention in the owner's manual being very careful not to bump the tip. This is critical. It is a very small, almost innocuous part of the device, but yet it's very critical. And when I handle samples, I always will put the sample to the edge of the stage and push it gently underneath. I never lift and I never push the sample down underneath the tip. I much prefer to rest it on the stage and slide it across. When you're mounting the chip, you want to make sure that it is seated squarely and that you have an even, firm tension with the screw holding it down. It's easy to tell when you go to tune up if the amplitude signal looks distorted or off center. You'll see similar distortions in the phase that you get as well. That's not correct. A simple loosening of the screw, a gentle shifting of the chip on its seat, and then tightening the screw firmly down, and you'll have a nice symmetric amplitude and a nice noise-free phase image when you have correct placement. There are 5 interactive parameters we'll talk about on the next slide, but in order to really be successful with the tool, you want to make sure that you're handling your samples and your tip with appropriate care. During tuning, you may find that when you first put a sample down, you'll see an image, but you don't get the highest resolution. Here we see a 1 micron region where if we were looking at this at a 10 times larger field, at a 10 micron by 10 micron field, you'd see a number of objects on the surface and you'd think, oh, this is great. But as you zoomed in, you can see that there's significant distortion. As I tune, going between the 2 key settings in the PID, the P and the I setting, I'm also being conscious of Z range, amplitude, and set point. These 5 parameters work together to refine the image. Going from left to center, this was mostly optimizing P and I. And then as I realized that the surface had changed from the previous sample significantly, I went back, I reset the Z range to be a little bit more appropriate for the height. This is a sample that's giving me almost 200 nanometers of Z height. So I shifted the Z range a bit. I controlled the amplitude. I adjusted the set point so the tip was pressing a little bit harder. And then again, adjusting the P and the I iteratively brought in this beautiful resolution. And you can see that this coating has significant topology at about 5 nanometers or so. And a truly refined version of this image we'll see later. The moral of this is that patient adjustment using these 5 settings will allow you to get true nanometer resolution as a matter of routine. This instrument has come to be a real workhorse for myself, and I've introduced several customers to this instrument as a standard tool. A word about graphics processing. Chin Shu showed some very beautiful multicolored images. There are several kinds of software that are available. Freeware called Gwydion does a remarkable job interpreting the files that the nGauge produces. But the nGauge itself generates a very high resolution image that you can take into graphics software. On the left, you see the image as it came off of the nGauge tool. And with graphics software built into my Macintosh, nothing fancy. I adjusted the brightness, a little bit of saturation, a little bit of contrast, and I was able to pull out some of the finer features that were quite hard to see in the original image. And with just maybe a minute or less of adjustment with a standard graphics package, I was able to really improve and make that figure pop out. So image processing with software really does do a nice job, not just making a pretty picture, but it will enable your analysis as well, because you'll be able to pick out features that you might not have been able to capture before with the measurements. So here are a couple of optical metamaterials. On the left, we have a single crystal of sapphire, upon which I etched 180 nanometer posts. And on the right, you can see a commercially obtained magnesium fluoride diffraction grating, a Bragg grating. This is a constituent of specialty paints. The individual particles themselves are on the order of 20 microns across. These are about 20 microns long and about 4 microns wide. In both cases, the ICSPI nGauge tool was able to quickly and accurately show me details of my surface. In the case of the single crystal sapphire, you could see that the interesting packing arrangement here is a minority compared to the hexagonal close packed, but this cubic close packed is really an interesting story all its own. And we're pretty excited to see that. In the case of the Bragg reflector, we wanted to ensure that we were seeing good quality materials, and we could criticize perhaps some of the roughness characteristics, but the spacing here was 555 nanometers per step, and the steps were angled at about 22.25 degrees, which was really well within the spec that we wanted for the application. We were able to validate in both cases that our prototype design parameters were being reached by the processes that we used to get those prototypes. And in both cases, the analysis was done in minutes, providing us absolute validation that we're on target with our design. Here are a couple of other optical metamaterials. In the left, we have a coating here reflecting reddish orange light, and it's transmitting bluish purple light under the microscope. You can see it's composed of spheroidal particles. These are hollow spheres of titanium dioxide and a titanium dioxide matrix. And we might be able to criticize some of the cracking behavior, but it's very hard to see details of what actually the spheroids look like or what their arrangement is. In the nGauge, it was trivial. We can see that they're slightly elongated spheres. The cracks are clustering behavior among the spheres, and we can then start to understand a lot more about how the surface relates to the optical properties that we saw, including, for instance, how the yellow fades to red as you change your angle of incidence, and the fact that we did have significant defectivity. In fact, the material worked quite well for our purpose, and we were able to use a very low-cost coating method to get this performance. In the right, we have a graphene oxide silicon dioxide composite that was spray-coated to make a similar inverse opal. These are hollow spheres of about 90% graphene oxide with a little bit of binders silicon dioxide. The topology of the titania surface is about 1.33 microns, and we're reading half micron sized spheres with approximately 20 nanometer resolution. You can see easily the surface roughness of those spheres. In the case of the graphene oxide, we've got about 4 microns from the deepest pit to the tallest peak, and we're able to see some of the surface topology in this image for those spheres. And again, these spheres are about half micron, and I think we're getting somewhere around 35 or 40 nanometers of resolution, despite that extreme topography. Once again, we're validating the prototype design specs were met, and we were able to see that our low-cost coating solutions and our chemistry were doing what they were exactly supposed to do. We're going to shift to metal oxide semiconductors. These images are both from a metal oxide coating on glass. Something that I wanted to point out with semiconductors and conductive materials on non-conductors like glass is they tend to charge. Charging can be a source of noise, and it's very simple and low cost to get a fan or there are a couple of handheld guns, and I have a little plug-in-the-wall tool that has alternating pulses of positive and negative electricity that'll neutralize surface charge. That's really helpful for working with conductors and semiconductors on glass. Very often in the semiconductor world, you'll be looking at silicon on insulator or other semiconductors on insulator. Certainly in the solar industry, we're engaged in making thin film solar, so glass is our substrate. With charge control on the surface, we're able to see fluorine-doped tin oxide all the way down to the individual facets and defects in the crystals, small amounts of secondary crystallization on the surface that was then used to be coated with a nickel oxide. And the nickel oxide you can see is covering over some of the topography, and yet you are able to see again some of the facets showing their nature, and we could diagnose both the surface quality and the coating consistency of these substrates. And we'll be looking at some other related materials, and you'll see exactly what I mean, because not all nickel oxide coatings are as uniform as others. Again, I stress that being able to examine quickly and efficiently going across many different kinds of materials is very important when understanding incoming material quality, process quality, or comparing one process to another in terms of coverage or surface consistency. Here we have a zoom in of those two same samples. Here we can see crystal facets in the fluorine-doped tin oxide down to just about a nanometer. This nice crisp line is a ridge. This nice crisp line here is a trough, and then here is another ridge. And if we look over at the nickel oxide coating, there's just a slight suggestion of the trough. We can see the junction at the edge of the crystal face here, this facet. But for the most part, a lot of the most concerning sharp topography is covered, and we believe that this is a contiguous coating. However, as I said, there are other ways of creating nickel oxide coatings. On the left, we see a different nickel oxide that has a very dramatically different surface. And I have to admit, I was surprised when I started to tune the instrument to this surface. The results, at first, I was hesitant to believe that I was looking at a porous material. Nonetheless, yes, indeed, you're looking at what look like porous flakes on this surface. Those conditions were somewhat unusual, but you can see some of these pits are really quite small. The smallest of these is down around 20 nanometers. And this particular image, we were able to claim something like 10 nanometer resolution without too much difficulty, even though the surface itself was quite challenging. On the right, you see a very different nickel oxide on FTO being put down, and a much more complex topography as a result. Although both of them have relatively similar peak to valley distances, the overall shape and nature of the surfaces are quite different, and you might expect that they might have different optical and electronic properties as a result. There's just no substitute for resolution, and the resolution from this tool is exquisite. This is a picture of perovskite semiconductor. This is a hybrid lead halide perovskite, and we're looking at the nature of the crystallization. On the left, we have a 10 micron by 10 micron field. In the center, we've gone a little past halfway. It's a 4 micron by 4 micron field, and you're beginning to be able to see some twinning in some of the crystals, understanding of some of the facets. But then when we go down to a 1 micron by 1 micron field at the junction between 3 crystals, you can see very clearly the nature of the junction, the fact that we've got definitely twinning happening there, and a good image of what the pit actually looks like, and that it is a fully contiguous coating, that the dark regions are just quite deep, but there are no pinholes, and it's a truly beautiful, well crystallized surface. And I suppose the moral at the bottom of this slide has been repeated, but it stands repetition. There's just no substitute for resolution. And along with resolution, you can also look at distinctly different chemical environments. This is an indium tin oxide surface that has relatively flat topography, at least at this scale. We're looking at a 400 nanometer by 400 nanometer field size. On the right, we have offset. That's just, the topography, more or less the kind of imaging we've been looking at mostly so far. And on the left, we have a phase image. And as Chen-Shu pointed out, the phase image can give you an understanding of differences in hardness that can relate to chemical composition. And, very clearly, even though in the topography, we see a step. This is actually a crystal grain merging into the next crystal grain over. But in the phase image, you can see that the crystal grain on the right looks somewhat different than the crystal grain on the left. And we believe that there is a compositional shift. What I really can't go into is the nature of the material that's sitting on the indium tin oxide, but I think you can see that at this resolution, there is definitely a real significant change in the nature of this film, and I believe we're looking at compositional variation across the surface. The step height here that we're detecting between the yellow and the dark red is about 2.8 nanometers. And so, as Chen-Shu noted, you can get sub-nanometer resolution if you try. And in this case, I think you'll see that the color gradient supports that quite well. In this 400 nanometer by 400 nanometer field, we're able to pull out, truly remarkable detail in terms of the surface quality and the nature of this complex material. By and large, I don't think there is a better, more versatile AFM instrument out there. I've been working with this tool for 8 years now, I guess. I met the founder at a conference several years before that, and I was very pleased to be one of the earliest users. And I just, it's a joy to work with. So, conclusions. The AFM, in this case, the nGauge, can be an incredibly important adjunct to your research and development or your quality control, quality assurance in production. And the ICSPI nGauge is really a cost-effective way of getting the resolution that can answer critical questions. Advanced optoelectronic materials rely on small-scale structure. When I first started working with the instrument, a good computer chip might have transistors around 20 nanometers. These days, we're looking at 2 nanometers. Some of the materials that go into building those transistors are 10 times smaller than that. The AFM is one of the only tools that can answer questions about surface quality, surface consistency. Many of the materials that I've shown you tonight, perhaps you could do SEM on, but on plastics and composites, and biological samples, that's not possible. This AFM technology, especially in such a small and affordable package, allows you to answer questions that really can't be answered any other way. Topology is critical for integrating materials, and the AFM is a key tool for answering these topological questions. The nanometer resolution is very straightforward with proper setup and tuning. You too can obtain truly exquisite results with this technology. Thank you very much for your attention.
Thank you very much for your insightful presentation and your talk today, Rob and also Chen-Shu. I hope all of us have idea how to investigate our material much better in nanoscale with this disruptive technology. I see some of the questions coming up. Maybe Xinshu or Rob can answer this. Question number one, for insulating substrate or polymer coated substrate, is there any indicator of the charging issue during the scan? I understand that we can see it during the approach step, not snapping in, but I wonder if we can also see it during the scan as well.
I think, yeah, maybe I can answer that question. Yeah, you can see it during, there's essentially, it's a little bit hard to describe. Like, it's best to show like an image. I don't really have a good image to kind of show right now.
I think I have
You have
one for you.
Okay.
It was one of the higher resolution images. I struggled a little bit with this. Hopefully it'll come right up here. If you look very closely at this pattern here, there are these faint wavy lines. I couldn't quite eliminate all of this surface charge. It kept building up on the sample as I was acquiring it. And I definitely have found a better way, but Chinshu, you might remember I was using one tool and I went to a different anti-static tool.
Yeah.
This was one of the reasons why, but it's these wavy lines that I was combating.
Oh, can you share your screen? We can't actually see it.
Oh dear, I'm terribly sorry. I thought that I had. Yeah, if you look at the high resolution image here, there are very faint parallel wavy lines along there that I've come to associate with charging. This is hardly any at all, but this sample certainly presented a particular challenge with this, and I was not able to completely eliminate it at the highest resolution.
Yeah, sometimes as well when you are scanning and there's charging, you won't get an image. It's basically going to look just like kind of fuzzy, even though you should be seeing something. That's like an indication of like you have very serious charging. I don't have a good image of that really to show right now. Oh, maybe I do. I can pull something up here. But let me see here. Do I have a good example of this right now? Okay. Yeah, okay. Here we go. This can be a good example of charging. I'm going to see if I can share this image here. I'll throw it into the chat if that's okay. Okay, I can't throw it into the chat. Trying to see. Okay, I can do this actually for people. Maybe not. No.
Okay,
okay.
there is actually another question. Okay,
Yeah,
you can answer.
Yeah.
Yep. This is from Philippines. May
Yep.
I know if AFM is capable of mapping out thin materials like wires that essentially have no support underneath them?
Yes, definitely. So the main thing with this is that you want to make sure that you're actually landing on the wire. So we can land on human hairs, actually. So the AFM is actually very precise. The main thing you want to be aware of is that you want to have like an actual microscope. So this is going into a little bit of the details between our nGauge AFM versus our Redux AFM. Our Redux AFM still uses the same core chip technology, but it has extra features thrown in to make it more user accessible. It has a built-in integrated optical microscope along with like a motorized XY stage. So it's very easy for you to land on like specific wire samples, for instance. You just have to make sure things are aligned properly. You can also do this on the nGauge. It also has a stage, but the stage is manual, and you have to set up a separate microscope. But yes, it is very much possible to do this.
All right. Thank you so much, Shenshu. And thank you so much, Rob, for sharing your experience. So I would like to share the survey here because your feedback is very valuable, dear guest. Please take a moment to scan and complete the survey so we can continue improving our webinars in the future.
I do believe we have one more question here.
Yeah.
From
sorry.
Thailand.
Is there? I know.
Yeah, I can see the question. I can read it out loud. Yeah. From your experience, how do each of the 5 tuning parameters affect the image? How do you adjust these parameters, such as which direction? I think you actually talked about this a little bit in your presentation, Rob, so you can feel free to talk about that.
Sure. I would say the first step is to get some kind of an understanding of the topology that you're working with. Understanding, roughly speaking, what size range between the valleys and the peaks. It matters if you've got, let's say, 2 microns of step height versus 100 nanometers of step height. That allows you to pick a reasonable value for the Z range. For a very small step height, I will tend to use a smaller Z range. And for a very large step height, I'll work with a larger Z range. I will then tune up normally, and that will help get me an initial set point. The set point is the amount of pressure that the tip exerts maximally when it comes to the bottom of its stroke on the surface. That's useful. The tool will automatically come up with a default setting. And as I then start to tune the P and the I will set the P just below the point where you'll see a feedback signal. It looks like very large scale bands. And if you back the P off and allow the I to come up, you'll see the image get crisper and crisper. And at some point, you'll reach an end for being able to optimize that. If you gently change the set point slightly, you can reduce the set point to increase the pressure. And very often, that will then mean you'll have to decrease P and decrease I, because that'll induce a little bit of feedback and you'll see the banding again. So what I will do is I will decrease the set point, decrease P and I, maybe decrease P, decrease I, decrease P, decrease I, and then bring the set point down a little bit. At some point, you're going to have to make a judgment call whether you're getting additional benefit or not. And usually after a couple of small adjustments, maybe 10%, 15% of the set point, I may try reducing the amplitude or increasing the amplitude to see if that can help. Very often, if I'm working at a very, very small resolution, trying to fish out a 5 nanometer width difference, dropping the amplitude by about 15% from the middle of the range, which is 2.5 volts, not to get too technical here, but if I go from 2.5 volts to 2.1 volts, then I might be able to get slightly better resolution. But again, I'll have to redo P and I, might have to increase the tip pressure a little bit or back off a little bit. And at that point, I can probably reduce the Z range just a little bit more. So it's, incrementally going back and forth across the set. And you don't want to go in order. You want to go like P and I and then set point, P and I and then amplitude, so that you get a uniform optimization. And often I decrease gently as I go. Does that make sense?
Yeah, that's great. No, that's a good explanation. Thank you, Rob.
That's a very nice explanation. So yeah, next, please take a moment for all of you. May you do the survey here by scanning the QR code presented on the screen now. And I would like to thank you everyone for joining and engaging with us today. We appreciate your participation and hope you found this session helpful. And we hope to see you again in our next webinar, and have a good day.
Have a great morning, everyone. Thank you so much.
Thank you,
Take care,
everyone.
everyone.
Thank
Yeah.
you.
Take care.